Hello, my name is Beth Fawcett, and today I would like to discuss with you a project that I've completed. And the driving question for my project was, with current gene mapping techniques, can I predict the location of a gene based on its composition? A little bit of background about myself. I am a chemist, and I'm really interested in proteins, so I wanted to develop a study about proteins. Now, proteins and genetic information are linked. The way that I like to explain how genes work is to picture beads on a necklace. So the entire necklace is DNA. And on a necklace, you can have multiple strands. Those various strands would then be the chromosomes. And then the genes would be the beads that are on those various strands. Now, genes have a location information called a locus, the plural of which is loci. And in those genes, you can think about those genes those particular locations, that particular bead might have some sort of faceting, some sort of cutting in them, and that would be like the proteins that make up that part of the bead. And for the purposes of this discussion, I have two different studies. One of them is about the location information, and one of them is about the strand. Now, the positive is from basically, you can think about it from moving from right to left on the gene, and then the negative is moving from left to right. So, both ways, whenever you're reading the gene information, can produce different types of proteins. So, the study that I have tried to classify the location and which direction the protein is being read in. The reason why this study is important is because knowing the location of a gene based on its genetic information, or the location of a gene based on its amino acid composition, can help us with drug targeting. And that's really helpful whenever you're working in drug development. Also, if we can access the location of a gene, then we can develop individualized medicine, again, through this type of targeting based on amino acid composition. And also, the amino acids are like the building blocks of the proteins that are made from this genetic information. So if we can understand their structure, maybe we can understand the mechanism behind some disease processes. So basically, there's a ton of different types of health applications, and the applications end where your creativity does. For the purposes of my study, I used a supervised learning technique, which basically just means all of my data was labeled. So whenever I went into the study, I didn't know. That's wrong. Whenever I went into the study, I knew exactly where that gene was located, and then I let my model kind of predict it, and then I cross-checked in between them. I used loci with more than 50 occurrences only. That just helped with my data integrity, and I ended up selecting chromosome 19, which had 48 different genes that had more than 50 loci on a particular chromosome. I mentioned I have two studies, the locus study, which is the location of the gene, and the strand study, which is the direction it's being read in. And the big thing that kind of drove me to think that this might be possible is that there's a relationship between protein and molecular weight. The visual that you're seeing is from the data that I personally used. This is a commonly known fact that as the length of amino acids increases, so does the weight of that particular protein. In particular, I found that tryptophan, which is a very heavy amino acid, was found frequently. Also, methionine was also found frequently. The reason why methionine didn't really surprise me is it's kind of like the start signal for every protein code. Versus tryptophan, I was really surprised to find it so prolifically throughout all of the samples. And again, it's very heavy, so that kind of drives this idea that there might be something related between amino acid composition and location of the chromosome, because if you have this big bulky tryptophan and you have a ton of it, you have to organize it in a way that it'll actually fit inside of the container being the nuclei inside the cell. I also found that cysteine and tryptophan's weights were really high for how commonly they were found. So tryptophan's heavy. I was surprised to find that cysteine was found as well. So cysteine wasn't as common in number, but whenever you account for weight, cysteine kind of popped out as one of the driving forces behind this weight length ratio, this the building blocks that make up this house that ends up being this protein. And so that those visuals and those graphs are kind of like the support behind why this study might have some sort of association. And what you're seeing here on this slide is actually the results of both the loci study and the strand study. So in the locus study, I developed a random forest model. You're seeing the results of over 200 inputs in my test cases. And what you're seeing on this diagonal line are the proteins that were fed into the model and predicted correctly. The only two that weren't are this one here and this one here. So those would be something that we call false positives. So they were positively identified, but in the wrong class. There were only 2 out of 200, and that's what drove my testing accuracy to 99%. The test accuracy for the strand study was not as great, but again, the same information is being fed in both forwards and backwards, so it makes sense that there's some kind of confusion there. And there probably is some adjustments that can be made in some future work. Speaking of future work, there are some better classification systems that I can use to drive that strand study to be more effective, specifically SVM modeling and using an XGBoost. I would love to expand. So right now, this is only for human chromosome 19. I would love to expand that to all human chromosomes. I would also love to use a neural network to possibly get some better information whenever I move to additional chromosomes. If you would like to contact me, this is my contact information. You can send me an email. You can look at the data and the code that was used to develop all of these things at my GitHub. Thank you for your time today. I really appreciate you stopping by and watching me explain my protein model.